Hi, I'm Suzie Alarcon. I am the NGS Core Manager at the Jolla Institute for Immunology, and today I'm going to talk about an assay that we developed in the Institute. It's a saliva-based RT-PCR SARS-CoV-2 detection assay. I'm going to talk a little bit about the deployment and the development of this test, or screen. I'd also like to thank the organizers for inviting me to do this recording today. So in summary, it's a one-step RT-qPCR assay. It uses the CDC novel coronavirus real-time PCR diagnostic panel. It effectively detects SARS-CoV-2 particles from saliva samples. Heat inactivation, together with direct lysis with detergent, actually eliminates the need to extract the RNA. We've got it down to a 6 microliter low-volume LV reaction, and that performs well. Just going to talk a little bit about the normal test life cycle. So in normal times, we start with the feasibility and design of a test, move into development, sketch out the validation, all the equipment, matrices that need to be tested. You do preliminary evaluations, and then you verify, and then you're ready to launch. You keep it maintained until it's ready for retirement. However, during a pandemic, this is the test life cycle pandemic edition. So although I put the talk together in a couple different ways, the way I'm going to go through it is more or less chronologically. So I'm not going to talk about, for example, stability and go through every iteration of every stability test at once. I'm going to take you through more chronologically just to highlight the way that development can be quite different during the pandemic and when resources are not as readily available as we may be used to. To borrow a phrase from Lisa Covington from the XPRIZE, which I'll talk about a little bit later, she said, we're building the plane while we're flying it. So that's sort of what this felt like. And I just kind of want to go through that, and because it was such a unique time to be designing something. The format, I'll just introduce each stage, define the objective, and I'll give a results summary, a TLDR, too long didn't read. The methods I'm pretty much going to breeze through. If you have any questions, you can look me up, and I'm happy to give any protocols or details offline. And then I'll go through the detailed results where I'll show the figures. The objectives that I'd like to convey through this talk will be to describe the advantages and disadvantages of a saliva-based assay, of an RNA-free, extraction-free assay to detect upper respiratory pathogens, specifically in this case SARS-CoV-2, and just talk about the flexibility and agility of PCR-based protocols. Some of the more common abbreviations I'm going to use, some of the more important ones would be the N and the N1 and N2 is the CoV nucleocapsid. RP is going to be human RNAseP, so that's going to serve as our human control. LOD, limit of detection, and the rest are pretty normal. NAN, not a number, so that shows up in some of our PCR results. And so why are we, as an independent academic institute, deciding to put a screen together? Well, as home to some of the world's top immunologists, it wasn't really an option for us to shut down, so instead we had to double down. Researchers at LJI contributed to some of the most important COVID research throughout this entire pandemic. So we, the NGS Core, were challenged to develop and implement a screen that would be simple for our employees to do on a regular basis and ensure they could keep coming into work and doing the important research that had to be done. We needed to balance sensitivity with ease, and the saliva-based assay fit that bill. This is Dr. Fauci highlighting some work from 2 of our researchers, Dr. Sete and Dr. Crotty, on T-cell immunity. So this work was informing policy as well. So I'm going to start at the very beginning, the proof of concept. The objectives were just, would it work? Could we take the CDC probes as they were and the IDT nucleocapsid positive control and just get it to work in the assay as we had sketched it out? In the results summary, yeah, it worked. The NPC didn't amplify. The COV did amplify in the positive RNAseP we found in normal saliva. So we figured out a few things. One is all of these probes work. Keep in mind at this time, all the RUO and the EUA versions of these probes and primers were all fan-based. So this couldn't be multiplexed quite yet. We tried 2 concentrations of Triton X as the detergent to lyse the saliva. We didn't attempt to extract the saliva at all. we went right into a direct lysis protocol. And if that didn't work, then we would have stepped backwards and looked into extraction. However, we were surprised, well, not surprised, but pleased to find that it did work quite well. This is just the plate layout of the titration that we began with the positive IDP control of the nucleocapsid, the plasmid. H2O is NTC, and then 3 presumed normal saliva samples. So these were saliva samples that were presumed normal and that they hadn't been tested in a clinical test yet to show that they were truly negative, but we just had to presume negativity until we could show it more through the assay. So I'm going to go through the slide here. This is the titration, the beginning titration of the plasmid control, and this is just NTE, and then 3 normal saliva samples. So we did not detect any COV in the saliva. The human internal control worked quite well, the RNAseP. In all 3 of those, the NTC didn't amplify anything. All the plasmid did amplify the plasmid, so we're good to go. We want to move on to the next step. So in that, we had tested 2 different Triton percentages for lysis. There was a paper out that had very few samples, but they used they think they used 20%. We wanted to see if we could reduce the amount of detergent that would end up in the assay. So this is a table, just looking at these numbers in the box, you can see that the negative numbers are more prevalent, and negative CT value indicates earlier amplification, because this was 10 minus 20, the CT values for those. And since less detergent is preferable, we proceeded with the 10%. Looking forward on this, we know already that saliva collection is going to need to be standardized. We're going to need to write great directions to have people be able to give a quality sample every time. So we wrote down very detailed instructions, and the team recorded a video that was available on YouTube for all our participants to be able to watch to get a better idea of how to give a quality sample. Just a graphic representation, we collect saliva through passive drooling just into any clean consumable works. We include in the saliva kit a transfer pipette, an empty tube, and then a tube that's filled with 1.5 ml of TE. So what we ask the participants to do is to actually make a working stock for us. This solves a few issues. One, saliva is viscous, and trying to pipette saliva, neat saliva on a liquid handler is quite difficult without dripping. Since we had a population that was very willing to test for us or to screen and follow directions, this was kind of an easy ask. If the population is different, then we've come up with other ways to collect the saliva without involving the participant making their own dilution. And then we ask them to clean off the tubes for us before they hand them in. One thing we want to do is multiplex. So as soon as we had the data showing that the proof of concept was good, that these probes would amplify, I ordered some custom probes on two of the targets to make sure we could actually amplify everything in one well. So we performed a 24-hour saliva stability just to see what kind of temperature we could keep the saliva at once it was brought into the lab. We did a 2-day process. On day 1, we tested them fresh. Day 2, after storing 3 aliquots of each of those fresh samples at 4, -20, and -80, we did the qPCR assay in quadruplicate again. And what we found was that all storage conditions were sufficient. They returned a good result for the RP at all conditions. The -80 condition had lower CTs in general and less deviation, so we stuck with the -80 and asked people. You can see in this box, the yellow is the fresh. These obviously weren't on the same plate because we can't test the fresh and a 24-hour condition at the same time. So again, we decided to store the samples at -80. We would like to test this longer and higher temperatures. We began a plasmid LOD to see if we could get through, see if we could determine just a limited detection of plasmid NTE. We also wanted to do a quick Thermocycler equivalency test to make sure we could use any machine in the building. So we did another COV plasmid titration, NTC, and saliva. So what we did was make a double volume plate and then stamp out one of the plates into the other one. This served to, this plate was both the COV plasmid titration and Thermocycler equivalency test. So it kind of served 2 things here. Again, 10, we can detect pretty reasonably. We have not reached the limit of detection yet. The average CT overall was lower on the Biorad. The standard deviation was lower. We're going to go with the Biorad and use the Jenna as a backup. This is just the method of the LOD. We still don't know the LOD. To find it out, we're going to have to put it in saliva. At this point, we get 4 clinically validated SARS-CoV samples in-house. And so we can finally move on to testing the assay with real positives. This is just a plate layout of some of the clinical positives. So we tested them neat, 50% for some of them because we didn't have enough volume to test all of them, and 25%. I'm not going to present the 50% data since I don't have it on everything. The way this is laid out here is samples 3, 4, 5, and 6. I showed the neat and then the 25% dilution and how the assay functioned. You can see sample 4 doesn't perform very well. Sample 3 is just okay. The 25% perform better overall, so we realize here samples need to be diluted. So we need to optimize and continue working on the clinically validated positives. Hopefully we get the multiplexing. The multiplexing probes came in soon after this. So what we decided to do is try this first with the plasmid and not waste our precious clinical sample until we know we can get it to work. So what we did is just compare the original probes that are all FAM, and we ran the multiplex version as well on the same plate. The open boxes are the original and the closed boxes are the multiplex, just to make sure that there was no sort of inhibitory competition between the primers and probe sets. Everything works well together, and in some cases better. This is just a table of those results. I should mention that this was done on residuals from all 4 of those first clinical positives. We didn't have enough volume left to run multiple reactions of anything on 1 or 4 samples, so we combined them all together and called it a clinical aggregate and continued some testing on that. This is the clinical samples, so everything performed well in the multiplexed reaction. So this gives us good confidence to move forward with this design. So we're going to continue to optimize the probes. Hex is the one that I picked for N2. It's kind of a noisy probe, so later on we decided to change it. We want to look at the stability of the positive saliva now. So we've investigated regular saliva and looked at the RP, but now can we still detect CoV particles? We assume yes, since the amplified regions in the N gene are so small, it would probably take a lot of degradation for this to happen, but we're not putting any stabilizers or preservatives in the saliva collection tube, so we just need to be sure that this is something that will return a positive result. So we go 4 hours and 24 hours. We have 2 new clinical samples now, 8 and 9. This is just a graphic layout of what we did. So there's 8 and 9. They were neat. They were made into 3 aliquots each of 25 and 12.5%. Those were tested right away. At 4 hours later, they were tested again, and then 24 hours later, tested again. And I broke it down by the percentage of the dilution. So here you can see sample 8 performs very well in all conditions. Sample 9 was viscous, and we learned that viscous samples, again, just do not perform very well. And this is going to inform our acceptance criteria for samples coming into the lab. Something that looks extremely viscous and we can't dilute down, we will probably reject and ask the person to hydrate very well and try again to give another sample, because hydration can have a big effect on the viscosity of the saliva sample coming in. This is a 12.5% dilution. Again, sample 8 performs really well. Sample 9 has a little bit of amplification here and there. It would have come back as an inconclusive is how we would have called this. The 12.5% didn't help too much, so we don't think that that's a viable option for extremely viscous samples. At some point, dilution doesn't seem to help all that much. After, if it fails at 25%, it's probably also going to fail at 12.5%. Is the major point of that. This is just sample 8, looking at the numbers, the 25% versus the 12.5%. So you can see all the data together. There's fresh 4 hours and overnight. The darker the blue it is, the more sensitive, the lower CT. So you can see there's not a huge difference here. The 25% works well. Fresh 4 hours and overnight all work great. Again, so we know that the saliva samples can be stored at least for 4 hours, which mimics the scenario that we're expecting, which is collecting at home and coming into work. Even if you get stuck in traffic, 4 hours should be enough. So another thing we wanted to do is test heat and activation. Because heat has been shown to effectively inactivate potential SARS-CoV-2 virus, we wanted to make it safer and sort of increase the logistical needs of the lab, so it would give us more flexibility in the staffing and the space and the automation. So what we found was that heat inactivation actually does not impact the detection. In some cases, it enhanced it. So we'll continue to implement this step upon receipt of all samples and investigate the stability before and after heating, which we do next. The viscous samples actually performed a little bit better after heating, so this is one thing that kind of improved the assay a bit. So this was sort of a crazy matrix test. We tested all kinds of conditions that we might see in the lab, which is if we heat both the stock tube that they hand in and the dilution tube that they hand in, and later we need to make a dilution from the heated stock tube and then heat that, does everything work? And does it work if we only use Triton X? And does it work if we only heat it? So all of these things were tested on a plate. There's no way you can see these numbers, but I just wanted to show the matrix of this plate and how detailed we tried to get to imagine every scenario that might happen and make sure that it was a valid method. So as you can see here, the gray box is the Triton X only. This is the protocol that we had been running. The light blue box is the heated dilution. The light yellow is a heated dilution and then lysed with Triton. The dark blue is where we heated both the stock tube, made a dilution, heated the dilution, and then the dark yellow is where we heated the stock, made a dilution, heated the dilution, and lysed it with Triton X as well. They all look okay. That's the end of that story. So we decided to move forward with heating just to make things a little bit safer, and it does actually improve the sensitivity in some cases. Next thing we want to do is evaluate the volume titration. So can we bring down the volume to not only save cost, but optimize throughput? So we looked at what we're doing now is a 20 microliter reaction. We brought down a 10, a 12, and a 6. I'll show in the table in the next how we did this. So the strategy here was to cut the volume of the final reaction, but keep the actual final neat saliva volume static over the two reactions. So you can see in the 20 microliter reaction, because we're using a 25% dilution and 5 microliters of that, it's 1.25 microliters of neat saliva. So say it's 1 copy per microliter of SARS-CoV-2, and then we're adding in 1.25 copies. Same thing with the 12. So we increase the amount of saliva being put in percentage-wise. We increase the Triton percentage, decrease the Triton volume. So the Triton percentage stays the same over, but we're able to add in more saliva, and we assume we're all adding in more copies of the potential virus per microliter. 6 microliter reaction worked very well, and this is what we end up handing into the XPRIZE, which is, I'll talk about in a little bit. I broke this one down by probe, so you can see N1 and N2 are the COV. Again, RP is the human internal control. 20 microliter average is the darkest one, and then 10, 12, and 6 is the lightest one. While the CTs are slightly higher in the 6 microliter for the COV probes, they're still well within a robust positive result here, and all of them showed up very, very well. 33 is about the lowest that we saw for a CT here. So I should note that in the extraction protocols, you may be seeing CTs in the teens, the 20s. We don't get that with the direct lysis. The CTs are usually in the 30s, sometimes up to 38, and that's a positive. So our cutoff is 40. Over 40 triggers a retest. Under 40 is a positive. So before we can test this in-house, we wanted to run some contamination testing just to make sure that moving the tubes through the building from the BSC to the liquid handler didn't introduce any contamination. We began with a checkerboard on just a pico green assay using DNA and NTC, just TE and water. Actually, let me go back here. So the top is what the adapters look like in the biomech. The first plate, we had the biomech dispense, and that plate got read right away. And then we ran the biomech through the qPCR program that would be running, and then we ran that through a plate again. I don't have the data to show for that because it was actually just photographs of the plate reader. but there was no contamination detected there. So we wanted to move on to an all-way checkerboard contamination test. So because in the previous one, an NTC splashing into DNA wouldn't necessarily show up, so we need to do this in an all-direction contamination test. So this here, you can't see, these numbers don't really mean anything except that the COV probes were found where we put the plasmid, and the RP was found where we put the RP30. There was one well, B10, that showed a small amount of RP, and it shouldn't have. So we brought the FAS in, they did calibration and alignment, we reran it, everything looked good, no contamination. So back to the plasmid and saliva, what is our real LOD here? Because saliva may inhibit, which we already saw, neat saliva inhibits detection of COV particles, we wanted to see if there's a threshold we can find where we can find plasmid in TE but not in saliva. It looks like it didn't really make too much of a difference, doesn't inhibit too much. So SP in this slide means saliva plasmid underscore and then the number of copies in the entire reaction. So SP1, for example, would be saliva plasmid with 1 copy spiked into a normal saliva. I should mention that we had pulled a bunch of normal saliva samples at this point to try and mitigate some of the between participant heterogeneity that can exist in saliva. On the right-hand side, you can see the TE underscore and then the number of copies of the plasmid that were there. So while we found positive results in some replicates, we didn't consistently find it until the 10 reaction, whereas in TE, we may be able to find it in 5. So we're calling 10 our limit of detection. I just wanted to point out here, this is just looking at 5 and 1,000 copies of plasmid. On the left is the plasmid in TE. In the middle is plasmid in 25% saliva. And on the right is the plasmid in neat saliva. So you can see in the 5 condition, it completely knocks out the detection of the plasmid. In the 1,000, it's there, but you can see one of the CTs is up near 40. So again, more data that shows that neat saliva can really inhibit this amplification. So this is just a graphic breakdown of how we end up. This is our final protocol. We collect the saliva, heat and activate it, dilute it, lyse it, add it in duplicate if there's room, run qPCR, and then analyze the data. And then I want to speak a little bit, on the variants and the probes and the agility that I spoke about in the beginning. So this is the nucleocapsid gene. This is adapted from Scripps keeps, they do a lot of routine surveillance, and they have this outbreak.info website. It's really cool if you want to play with it. So the yellow parts here are where our probes are amplifying. Those are the sections. You can see in the B117 at position 3 is kind of close to where the first probe is. However, we have tested a B117 variant sample through this assay and it still performs quite well. So what if a mutation comes up where one of our probes are? Well, it's not going to be a big deal really to design around it. We already have one in-house that we use as a backup. We've tested and validated it. And part of what's going to be cool about PCR testing in the future is keeping an eye on potential emerging pathogens. And I think next time, hopefully there's not a next time, but if there is, we would be more prepared and already have these things designed and potentially validate RNA or something like that later. So I wanted to go into the XPRIZE, which was, we were extremely fortunate to be able to enter and just sort of get access to a really cool network of people. This is the timeline, which you probably, again, can't see this here, but it started in July and then the final winners were just announced, I want to say March, maybe the end of February. So it was a lot of stages through, a lot of innovation that people presented there. So we formed a team LJI, and we decided to throw our hats in the ring. And through the XPRIZE, we were able to gain a lot more clinical validation of our tests or our screens. So in the semifinals, they sent us blinded plates. One interesting thing we found out is this assay is completely incompatible with PBS. We spiked plasmid in PBS. We were finding some weird results, and we didn't understand why we weren't getting any positives in certain plates, and then went back and looked at the matrices and our assay only worked on the saliva, and it worked quite well. We had a bunch of experiments planned to figure out why that was, but in the end, did not have the bandwidth to try and figure out why our assay wasn't working on materials that we didn't design our assay for. Well, it would be interesting. It wasn't something that we had the time to do. So in the semifinals, you can see our Sensi and Speci were 94 and 100. And then in the finals, there were 40 negatives, 40 positives. In each of these, there was one that we are calling a false negative. They were inconclusives, meaning it came back with one of the COV probes or three, but not all four replicates. So to be conservative, we're still calling it a false negative, though this would have triggered a retest in the real world. So in the end, averaging these two tests together, we have a Sensi and a Speci of 96.6 and 100. The reaction volume, one of the things that XPRIZE was looking for is that a test that's entered be radically affordable. So in a best case scenario, we would have everything designed out well. We have the reactions down to $1.22, which includes all the saliva collection and the batch controls as well. And we were excited to find out that we were one of the winning teams selected, and we are working on a deployment phase coming soon where we do a larger scale real world deployment. So we're super excited about that. So I just want to go through the objectives here, the advantages and disadvantages of saliva. So saliva, everybody's got it. It's easy to get from people. The quality remains good over multiple days. It's a stable sample, even though there's probably lots of RNases existing in there. Even after 5 days, we could still find everything that we were expecting to find in any sample. However, it can be difficult to get a quality sample. It can be viscous. It contains inhibitors. As you saw, a lot of the neat saliva inhibited any amplification of things. So that's one thing that can be difficult. The CTs are higher, though with the clinical validation, we learned that at least with those samples, it was still very specific and sensitive. So RNA extraction free. There was another team that used the same probes and primers, except they were using extraction. Their sensitivity came back at 100%, meaning they didn't have that inconclusive. However, this could be mitigated if we treated an inconclusive and reflexed it to an RNA extraction protocol. So I think that's something to get around. And again, the flexibility and the agility of PCR-based protocols is that if you can determine what genes are less likely to undergo a lot of mutations or variants and design your probes in there, that's probably a wise move. So I want to go through the acknowledgements, and I really want to thank Steve Wilson, who is my boss and our COO and was the brains of the operation and a hugest cheerleader for us. John Stillwagon, who wrote our entire reporting program and had everything working in a matter of, I don't know, weeks. And especially I'd like to thank the NGS core. This is my team, Arturo, Hannah, Kyle, and Rosie, who just worked like crazy doing all of the wet work for this and keeping the NGS core running at the same time, because that's our day job. I also want to go through and thank some other people at LJI. First, I want to thank the day porters who just cleaned and disinfected and deconned and just made it a safe environment for people to go into work. I know they worked so hard throughout this entire pandemic, and I'd just really like to thank them for providing a safe workplace for us. Ingrid, Chris, and Talia, who worked on the positive samples. Morag, Shane, Susan, Sonia, who helped us share their space and resources as well. David, Melissa, and Lisa, EHS, Biosafety, and IRB. John, Deanna, and Janae from facilities. And then I'd like to thank the XPRIZE team as well, Jeff, Lisa, and Chris, and then Brian, Dan, Karen, Kaden, and then our clinical partners who ran all this testing for us with almost zero training and just receiving a protocol and being able to run it was just amazing, especially Biosha. They were incredible. So I'd just like to thank all these people who helped us get through this entire process and just really made it, this wild ride, a little bit more fun. So thank you.